Jessie is 19 years old. She wants more information about birth control and her options to stay safe.
When she visits her doctor, she asks about the forms of birth control available to her. Her doctor tells her that there are misconceptions about hormonal birth control and that these misconceptions can cause long-term negative effects on the reproductive community. While hormonal birth control is primarily used for prevention of pregnancy, it can also be used for treatment of a variety of diseases or disorders, such as polycystic ovary syndrome, dysmenorrhea or menstrual cramps, and menorrhagia, or heavy, prolonged menstrual bleeding.
Different types of birth control include condoms, the IUD, the implant, the patch, oral contraceptives, and Plan B. Our project will focus on hormonal birth control, especially oral birth control and Plan B. Oral contraceptives, also known as the pill, are most commonly used in the US, and the pill is taken on a daily basis. These medicines can be delivered either as combination hormonal contraceptives containing steroids such as estrogen and progestins, or progestin only. The same hormones that are in various types of birth control are also useful in hormone therapy for transgender individuals, in treatment of hormone-sensitive cancers, in menopausal hormone therapy, in fertility treatments, and they're even being studied for use in male hormonal contraceptives. The hormones delivered via oral birth control are synthetic steroids, and they affect the action of certain receptors. Specifically, progestins, which are synthetic versions of naturally occurring progesterone or testosterone, are used in combination and progestin-only oral contraceptives, and they bind to the progesterone receptor. Progesterone is lipophilic and therefore can easily cross the membrane of a cell to bind to target progesterone receptors in the cytoplasm. The hormone receptor complex functions as a transcriptional factor. It is transported from the cytoplasm to the nucleus, where it can enhance or repress the expression of genes involved in making particular proteins, often affecting reproductive behavior. Progesterone binds primarily to the progesterone receptor, PR. The progesterone receptor can be found throughout the body in 3 isoforms, PRA, PRB, and PRC. It is one of the various type 1 nuclear receptors, and this family of receptors includes others like the androgen receptor and the estrogen receptor. These receptors tend to contain 3 functional domains. In PR, the domains are the N-terminal domain, the centrally located DNA binding domain, which is involved in DNA binding and in receptor dimerization, and the C-terminal domain, or also known as the ligand binding domain, LBD. This is the domain of note, where we have ligand binding, and this domain is involved in transcriptional activation. It contains 11 alpha helices and 2 short beta sheets. Progesterone binds in the active site, progesterone receptors undergo significant conformational change, which leads to their translocation from the cytoplasm to the nucleus and helps bind it to DNA. Progesterone, or other related ligands, bind to the progesterone receptor in the ligand binding domain, where it interacts with various amino acid residues via polar and hydrophobic interactions. These interactions help stabilize the LBD in its new active conformation. They do this in 2 ways: by connecting the progesterone to residues of the carboxyl terminal helix 12, and by stabilizing the lower part of the ligand binding domain, so that the helix 12 remains in this conformation once there. The interactions between the ligand and helix 12 are important as we consider which progestins can be used as contraceptives.
Because they are synthetically derived from naturally occurring hormones, progestins found in oral contraception have shapes that can interact with type 1 nuclear receptors that would normally bind to hormones like estrogen, testosterone, and progesterone. A commonly used progestin-only pill contains norethindrone. Synthetic hormones like norethindrone are testosterone-derived. Though their basic steroid structure is the same, there are a few key differences between progesterone and norethindrone. Unlike progesterone, norethindrone does not have a methyl group at C19. Also, C17 of norethindrone features both a hydroxyl and ethanol group, whereas progesterone features a ketone group. In binding to the progesterone receptor, agonists like norethindrone set off pathways that inhibit ovulation, make vaginal fluid thicker to prevent sperm from fertilizing an egg, and change the uterine lining to make implantation of a fertilized egg unfavorable.
On the other hand, Plan B one-step also delivers hormonal birth control in the form of a pill, but it acts much more quickly and drastically than do oral birth controls that are used consistently as medicines. Plan B was approved in the US for use as emergency contraception in 1982, and it can be taken as a single dose within 72 hours of unprotected sex or a contraceptive accident, but it does not end an existing pregnancy. As an emergency contraceptive, Plan B works quickly to temporarily delay the release of an egg from the ovary. By preventing ovulation, the medication decreases or eliminates the possibility of the ovum encountering sperm.
When the levonorgestrel in Plan B encounters the progesterone receptor, it binds in the active site as progesterone and norethindrone would. Much like the progestins found in the hormonal birth control pill, the levonorgestrel in Plan B is a synthetic testosterone that binds to the progesterone receptor. Like norethindrone, levonorgestrel acts as an agonist of the progesterone receptor. Its structure differs from that of norethindrone in only one place, where norethindrone has a C13 methyl group. Levonorgestrel is characterized by a C13 ethyl group. As we mentioned, these three steroids have remarkably similar structures. They bind in the same pocket of the ligand binding domain, but the small differences in their structure mean significant differences in their interactions with amino acid residues of the progesterone receptor. Here's a zoom in of the levonorgestrel with the amino acids it interacts with in the binding site. As a reminder, the ligand binding domain contains 11 alpha helices numbered H1, H3 through H12, and 2 short beta sheets. Numerous van der Waals interactions hold the levonorgestrel in place, but there are other notable non-covalent interactions of note in the active site. Some of these interactions also happen with progesterone and norethindrone, but some do not. For example, the C3 ketone on all three steroids is non-covalently anchored via hydrogen bonds to glutamine 725 and arginine 766, as well as to water.
So if these compounds all bind somewhat similarly, what explains their different uses? It turns out that the small differences in hormone structure can account for their different uses and their different potencies. An article by Petit Topin and others examined the differences between activity of naturally occurring steroid hormones and molecularly similar compounds. The authors found that residues of the progesterone receptor interact differently with all the three ligands that we've discussed, greatly affecting their activity. The difference in their interactions with these three residues is thought to be key in how they activate the progesterone receptor. The presence of a hydroxyl group at carbon-17 greatly enhances the potency of the progestin. Both norethindrone and levonorgestrel have this functional group, but progesterone has a ketone here instead. The hydroxyl group at carbon-17 is where norethindrone and levonorgestrel interact via hydrogen bonds with water and then to the residue of asparagine 719 on the H3 helix. At another residue, the absence of the carbon-19 methyl group bound to carbon-10 on levonorgestrel and norethindrone greatly enhances their potency as progestins. Progesterone has a carbon-19, which interacts with methionine 759 on the H5 helix. Because levonorgestrel and norethindrone do not have a carbon-19 at this particular location, their placement in the binding site is slightly different, permitting the steroids to interact more with the receptor. Finally, different structures at carbon-13 result in different interactions at methionine 909. Both progesterone and norethindrone have a methyl group at carbon-13 and contact methionine 909 of helix 12. Levonorgestrel, on the other hand, has an ethyl group here instead, and this allows methionine 909 of helix 12 to rotate away from the steroid, interacting instead with many other residues on other helices of the receptor. Researchers believe that this is the key difference accounting for the accelerated activity of levonorgestrel. The fact that levonorgestrel and norethindrone are only different in their carbon-13 substituents suggests that this is a key group allowing levonorgestrel to establish additional stabilizing contacts within the ligand-binding domain. Because of this difference, levonorgestrel, the more potent, can be used for immediate effect, and norethindrone, which has a strong but more moderate affinity at the binding site, is used instead for long-term medication.
Now that Jessie understands her options more, she can make safe and effective decisions for herself.